Hi, good afternoon. It's a pleasure to be here today in this session. But first, I will introduce myself. I did master and doctorate at Federal University of Rio de Janeiro. I'm currently a specialist at the Fiocruz Foundation, manages strategic projects in the area of technological development based on molecular diagnosis and automation, meeting the demands of the Ministry of Health. I have experience in developing and standardizing molecular diagnostic tests as NET, viral load, and type for HIV, HCV, HBV, malaria, dengue, Zika, chikungunya, influenza A, influenza B, and SARS-CoV-2. In this session will be panel 2, and today we're talking about the new diagnostic and epidemiological surveillance tools to deal with emergent viroses. We'll have 2 presentations, and the first one will be next generation sequence in epidemiological surveillance of emergent viroses and with emphasis on variants of SARS-CoV-2. And the second talk will be microarray based on based assay in diagnostics and epidemiological surveillance of emerging viroses, okay? It's a pleasure to introduce these 2 great researchers that we have here today. We will have here Ana Tereza Vasconcelos, our first speech. Ana Tereza is Senior Research Scientist at the National Laboratory for Scientific Computing and Coordinator of Bioinformatics Laboratory and Computational Genomic Unit Darcy Fontoura de Almeida at LNCC. Her team has experience in the area of bioinformatics and computational biology, using high-performance computing and IA, working on the following topics: genomics, developments of software application in bioinformatics and computational tools, apply assemble, annotation and comparison of genomes, metagenomics, exomes, and transcriptomics applied to the many different models organisms. Since 2020, she has worked in the generation and analysis of SARS-CoV-2 genomes, mainly in the identification of new lineage, okay? And our second speech, she has the last 15 years, Maria Luisa Villahermosa has led the development of more than 15 in vitro diagnostic products for the simultaneous detection and identification of a large numbers of microorganisms present in the human samples, endpoint mutations, insertions, and deletions present in human DNA and RNA. All developments have been carried out under the ISO standards to obtain CA mark correctly. The technologies used are based on multiplex PCR and detection through hybridization with specific probes located in a microarray, CLART technology of 2005. And during 2020, the latest products have been also developed in real-time PCR technology with the objective of detecting SARS-CoV-2 with the maximum possibilities that we have. During her professional career, she has led the development of new projects such as the concept of rapid PCRs, lyophilized formats, circulant tumor DNA analysis, liquid biopsies, and a new diagnostic tool based on point of care. Collaborations with other entities have led to the acquisition of a deep knowledge in the area of massive sequence with the analysis of exome and of wide panel of genes. And I would like to thank the speakers for being here today and able to contribute to this symposium, okay? This will be a small panel of 1 hour, less than 1 hour, a panel session with 2 short presentations for 10 to 15 minutes each, followed by 20 minutes of Q&A and debate sessions. The audience is invited to interact with us through the Q&A and the chat fields on the right side of the application. To start the session, I would like to invite Tereza to bring us the most recent information about the next generation sequence in epidemiological surveillance of emerging viroses with emphasis of variants of SARS-CoV-2. Please, Tereza, let's talk.
Hi, first of all, I would like to thank Dr. Akira to invite me, all the organizing for this symposium, to be here to present a quick presentation for what we are doing. I can share my screen? Yes. Can you, it's okay? You can see my screen? So I will start talking about, so I'm working at the National Laboratory for Scientific Computation, and I would like to present some work that we are doing at the lab, but we have to start to talk a little bit. The last survey from the WHO that presents the number of cases and the number of deaths for COVID-19 case report recently. So the number is growing. And then we can see that America is responsible for most of the cases at this moment. Brazil and United States are the highest countries that the most of the case, and now we are Asia that present a large number of the case and deaths at this moment. So the main objective for the SARS-CoV-2 sequence is to prevent to possibility the early detection and the characterization of the emergent variants and define if they are or not a particular concern. I will talk a little bit about that, but would like to know also the impact of these genetic and antigenic variants for the pandemic, and we have to monitoring them over the time and help to guide the public health systems. So we have an agreement to sequence and try all the countries all over the world to try to track in the spread the SARS-CoV-2 genomes geographically during the time, and to ensure that the mutation could be potentially influence the pathogenesis, the transmission, and the countermeasures such as vaccine, therapeutics, and diagnosis. The price of the sequence has dropped significantly last time, but we will still require equipment, reagents, and mainly the bioinformatics infrastructure to analyze all of this data. And the mondial community are encouraging the creation and the change of knowledge between the network that's all over the world. So we have two types at this time that's important, the variant that's very important. One of those that we are talking, one is we named it's the variant of concern, that is the variant that we have evidence that they increase the transmissibility of the virus and that they can cause more severe disease. This means that the number of hospitalization is increased and or the deaths are also increased when we identify these variants. And the other is the variants of interest, that we can see some genetics marker in his genomes that can be associated with the change for the receptor binding site to reduce the neutralization and to generate some concern about the infection and for the vaccination. So at the moment, all over the world are running to try to follow these variants. And up this moment, we have three main variants that we call the VOC, variants of concern. The first one that's appear in United States, the name is 1.7. And the other one is that appears in South of Africa, was identified in South Africa, the 351. And we have the P1 that has been identified here in Brazil in the north of the country. So the three variants arrives more or less in the same time, identified in the last September, in the last December 2020. And most of them, they have in common this mutation, this N4111 or 2, and some deletion in these genomes. These three VOC are related to increase of transmissibility and also increase of death, but it's not related for the vaccines, it's not to interrupt, prejudice the vaccine up this moment. And we also have another VOI that is a variance of interest that has been described in many countries all over the world. And some of them has been described here in Brazil, that's P2, that our group are involved in that. And now this is the variant, the 617, that has been the double mutant as the people are now talking about. That's up here, has been identified in India 2 months ago. So most of the variants is identified in the spike protein. That is the binding ACE in the human cell. And here we can see some from the England, from the South African, from Brazilian virus, that this mutation are the convergent mutation, this spike protein, especially in some sites of these mutations. Here is a world map that's very impressive, that we can see that the variant from the England that identified the 1.7 spread all over the country fastly. Here is the slide from the variant that's identified in South Africa, and this slide is from the last of the final April of this year, and he spread all over the country. The same thing. The same thing is about the P1 has been described here in the north of Brazil. Now he's spread all over the country. So we have to follow these variants and to identify there's appear a new one, or if those variants are still evolving, so still have more mutants in these variants. So we have to collect this. For doing that, we have to collect the sample to prepare the sequence, and then to analyze using the different bioinformatic tools, and then to share fastly in the fast way to public in the public database, and then to avoid the whole community to interpret this data, and also to produce the data for the public health systems. So as far as we know, all the vaccines continue to work to the three different type of variants at the moment, and also producing about the neutralizations is also work, one less or other not so good, but it is still working very well, the vaccines up to this moment. Here's in the fast panorama about the Brazil. Genome has been sequenced in Brazil. We have some states that we have more sequence that has been produced. We started the lab to sequence in the first wave of the SARS-CoV in Brazil. We start to sequence in the collaboration using the mini-ion technology. We produced 400 genomes with another groups spread all over Brazil. And then we started a genomic surveillance in the state of Rio de Janeiro, supported by FAPERJ in the Genomica network. So I will skip this one because I don't have much time. It's only to show that in this paper here, We are sequencing 200 genomes in the last December, and we are able to identify the P2 VOI, this variant of concern that we discover here first in the state of the Rio de Janeiro. We can see the clade here, the clade 2, and perform the phylogenetic network. We can identify this P2 that's start in Rio de Janeiro, then now he's spread all over the country. We continue to do the epidemiological surveillance in the state of Rio. This is a map what we had until the 2 of February of this year. And now we are doing a 400 genomes at each 15 days to improve the surveillance in state of Rio de Janeiro. This was the panorama until 2 of February. And now you can see that this changing in the middle of March. The P1 is dominate in the state of Rio de Janeiro. The P2 is now disappear, and in the last survey we finished today, we can see the P1 continue, but we discover in the last sequence that is still a new one, P1, that we call P1.2, that is changing the state of Rio de Janeiro. We can see the cluster of this new variant of the P1, the P1.2. And here is the mutation that we found in some samples. In some of them are in the spike protein. So this is the snapshot, the panorama that we have here in Rio de Janeiro up this moment. Here we can see at the beginning we have the P128, and the P1 33 that is dominant in the state of the Rio, and in the middle of the last year, the P2 start to appear here where we sequence and we discover the P2 in Rio de Janeiro, but now the P2 is decreasing. In the opposite side, the P1 start to appear, and now he's dominant in Rio de Janeiro, and now we have the new P1, P1.2 that appear to increase in the number here. So I think we have my time. I would like to thank you, the Genomica Network, the Secretaria de Saúde do Estado do Rio de Janeiro, the Prefeitura do Rio de Janeiro, the LACEN, and o Laboratório de Virologia Molecular, the UFRJ, and the FAPESH to support this work. And also we work in the Genomica for the Ministry of Science and Technology, and I would like to thank everybody that is involved. Thank you.
Thank you, Anna. It's an excellent presentation. And to continue this session, I would like to invite Maria Luisa to bring to us the most recent information about microarray-based assays in diagnostic and epidemiological surveillance of emerging viroses. And Maria Luisa is now with you. Maria, I can hear you.
Can you hear me now? Okay. Patricia, I don't know if you can hear me properly.
Yeah, now it's perfect. You
okay,
go ahead, please. It's excellent, now the sound.
Okay, thank you. So thank you very much for inviting me to this event, and thank you for giving us the possibility to show you our work. Sorry. Before to speak about the assays based in microarrays, I would like to tell you who we are. Genomica is a multinational company focused on molecular diagnostic. We design and manufacture in vitro diagnostic kits, and our products are based on our technology, which combines multiplex PCR with CLAR technology, and we have other products with real-time technology. And always we keep a high quality commitment. Regarding the CLAR technology, I show you the workflow of this technology. Really, the technology comprises the step 2, 3, and 4, because the step 1, which is extraction of the genetic material of the microorganism that are in the clinical samples, you can use some automatic extractor. These extractors are in all the labs, in all the centers, and works well with our technology. Regarding the first step, is the multiplex PCR. We design a multiplex PCR in order to amplify several targets in the same tube. We have products in which you can amplify more than 10 microorganisms in the same tube, and one of our advantages is that we can design this multiplex PCR very specific, and we can detect and we can amplify subtypes of the same microorganism, a microorganism with similar sequences. After this step, we have this second step, that this is the visualization process. In this visualization process, you have to add the amplified product to a well, and in this well, there are probes printed in the bottom of this well. I show you in detail in the next slide. Here is a well of the 8 strip, and each well means one sample. In the bottom of this well, we can print more than 100 spots. Each spot contains one probe, and this probe is specific for a DNA sequence. When you add the amplified product, you add here the amplified product, then this amplified product will hybridize with its specific probe. And after adding several reagents, you obtain a precipitate. You obtain a result like this, and in our reader, this reader transforms the microarray specific pattern into a clinical report by using a Genomica's image processing software. For example, in this assay, this is the virus which will resulted positive. The rest of the virus were negative. Regarding the equipment, all the options that we have allow us to perform up to 96 samples in one run, in the same run. Depends of the throughput that the clients needs, can select different equipment. Here you can see the reader in which you introduce the microplate and you obtain the report directly. In this equipment, all the visualization process is made in an automatic way. And in this last equipment, the AutoCLAR Plus, you introduce the microplates with the amplified products, and all the visualization step is made inside of the equipment, including the generation of the report. Why CLAR technology could be a good tool for epidemiological surveillance? We have three reasons. The first reason is the short time to develop a new target. The second reason is several advantages of CLAR technology that I will explain in the next slides. And the third reason is because it's easy to include a new target in a persisting CLAR panel for virus prevalence studies and epidemiology studies. Regarding the first point, an example is the last year we developed a product for detecting SARS-CoV-2. We developed this product in 5 weeks. In January 2020, the sequence of SARS-CoV-2 was published and Genomica develops the kit in 5 weeks, including the CE market. We analyze the sequences, design the primers and the probe, analyze the rest of the sequences of the rest of the coronaviruses. We did the optimization of the PCR and the visualization process. And all of this development was made using commercial RNAs because the samples were not available in that moment. And finally, the clinical validation was performed in a reference center of Spain, and because all the positive clinical samples were sent to this center. Regarding the analytical parameters, we obtain a sensitivity of 100% at 5 copies of the RNA of the SARS-CoV-2. And regarding to the specificity, we test several RNAs, including the different coronaviruses, the NL63, the 229E, OC43, MERS, SARS, and we added more than 1 million of copies of each RNA, and non-specific detections were observed, being showing a specificity of 100%. The cross-reaction study was performed with 76 positive respiratory samples with different microorganisms associated to respiratory infections, and no cross-reaction was observed. And finally, the diagnostic parameters were determined in a panel of 80 samples, was made in the National Center for Microbiology. The Institute of Health Carlos III is our Spanish National Reference Center, and the results show more than 95% of sensitivity and specificity. Move to the second point. I would like to show you the advantage of CLAR technology. One is the automatization. You can perform up to 96 samples in one run. The multiple detection and identification of several targets in the same assay, including the controls. No requirement of additional equipment. The PCR tubes are ready to use, no need to prepare master mix, and it's a fast and specific diagnostic tool, taking in account the throughput that the client needs. The third point is that in this technology it's easy to include a new target in a persisting CLAR panel for these studies of prevalence of epidemiology. We have a product for detection and identification of 21 respiratory virus types and subtypes, and all of these viruses are detected in the same assay, including the coronavirus, the non-coronavirus, the bocavirus, adenovirus, all of the virus that you can see here. And this year, we will launch this product, including the detection of SARS-CoV-2. We are including the 2 genes or RF, and every week we analyze the new sequences in order to verify that our primers and our probe continues being good for detecting the virus and the new variants. The processing time is 3 hours, up to 96 samples, including the extraction step, and an endogenous control and amplification control is included in this product. Finally, I would like to show you the different products that we have in the market in order to see that we can detect and identify a high number of viruses and bacteria. And I think that this product do CLAR as a good tool for studies of prevalence and epidemiological studies. Thank you very much.
It's incredible talks. Thank you all for the thoughtful presentation, and we invite you back to the screen to start the Q&A sessions. I would like to reinforce that we are receiving comments and questions from the audience in the event application. If you have any query to any of our presenters, please write at the chat or to the Q field to the right side of the screen. To start, we have some questions, interest questions, and the first one will be for Ana Tereza. We have seen the emergence of virus variants throughout this pandemic year. Do you have seen recombination events in SARS-CoV-2 that can create combinations between the variants, as we all see in HIV, for example?
Thank you for the question. I didn't have time to show, but we have seen co-infection. We described the first co-infection in Brazil in 2 patients from the south of the country. So we are looking also for the variants from that we call SNFs. That is a number of variants that can do the recombination inside the cells and then produce the co-infections in the patients.
Okay. And do you think that could be a huge problem, a bigger problem than we have today related to these variants of concern if we have this kind of event of recombination?
Yes, this is a huge problem that we have to look, and we have to sequence more with high, deeply sequencing to be able to identify this amount of the variants from that can arrive in the middle of the person. That's a huge problem. And as we have a lot of people we don't have a lot of vaccination in Brazil, and a lot of people that are not using the protection, etc. These events will worsen in the next months.
I'm really preoccupied too with this possibility. And Maria Luisa, is there a plan for Genomica develop a CLART COVID-19 for variants of concern? Would it be possible to design a product with the ability to detect new variants before genome sequence?
Really, sometimes during the last month, we have discussed in the company the develop of this kit for analyze for detect the new variants. The CLART technology is able to detect SNPs and deletions and insertion, because we have several products in oncology in which we detect these point mutations. Then it's possible to add these mutations in order to identify the new variants. But at the end, also we develop in 3 months these products, the variants appear as Anna show us in the before presentation, appear very quickly and change very quickly. Then in this moment we are waiting for decide this the development of this kind of product. We're waiting because appear new variant, appear new SNPs, then maybe it's not the moment for develop, because the CLART technology, if you can add SNPs, you can detect SNPs, but the time that you spend in develop the product, I think that surely new variants and new mutations could appear. But the technology, from the technical point of view, is possible to develop this kind of product.
Okay, based on this, Dr. Anna, do you think that it could be possible to predict a new virus mutation when we use this kind of bioinformatic structure?
Yes, first of all, I would like to agree that I don't think it's a good idea to the variant disappear in 2-3 months. So to invest a huge effort to develop a new test to identify one variant, I don't think the sequence test is now cheaper. So sometimes better to continue to perform the whole genome sequence, identify as much as possible the variants that appear. Yes, we are doing some of these analysis to try to identify the possible new variants using artificial intelligence methodology and processing a huge amount of data from the public database in order to identify which are the sites that is probably more susceptible to have a mutation. So we have a lot of groups all over the world, including my group at LNCC, that we are trying to work on in this field, and is feasible. Completely feasible. I think that's the movement that we have to do now.
And do you think that could be interesting to input with the data of the huge amount of vaccines that we are facing today, if the different types of vaccines could impact in this virus fitness, and based on these could predict better the new mutations?
Yes, this is a new for us because the people are starting to vaccinate. So we don't have a lot of the data that can support at this moment this type of analysis. So I think that as much as we have the clinical data and the epidemiological data from the patients, that we can possibly integrate this data in order to verify if the models that we are producing, all the groups, and aggregate the information about the vaccine and the mutants that will arrive, because they will arrive some virus that will be resistant to the vaccines, and then to improve the models.
Yeah. Maria Luisa, do you think that relates to the challenge that is to develop diagnostic kits for this moment that we are living, in pandemic moment that we are living? Do you think that, what do you think that could be the most important challenge and difficulties that you and your group faced during these moments that you are in charge, the company is in charge to develop diagnostic kits?
Sorry, I'm not sure if I understand exactly your question. Is that what is our?
I try to reformulate my idea. The COVID-19 pandemic has faced us with several challenges and difficulties. In your opinion, what is the greater learning so far that this moment has taught, and that you and your team will take to face other emergence virus?
Okay. During this last month, we have learned several things. We have collaborated a lot in research centers in Spain and out of Spain, and this is very good. And we have we have developed several improvement for CLAR technology that maybe before the pandemic, before this, we cannot think with superiority. One of the things is to develop the product without the extraction step, for example. Now we have the product. We have worked to remove the extraction step with very successful results, and I think that it has been the SARS-CoV-2 tell us, okay, there aren't enough material for extraction in the world, and this is one of the things that we have learned during these days. And I think that the things go faster than before, and I don't know what more, but I think that we have learned things to collaborate, to do things very quickly, and to think in a global feelings. We have in collaboration with a lot of companies in Spain too, because, for example, other problem that we found during this last month was the reagents as oligonucleotides. It was impossible to have oligonucleotides contained in the company. I don't know, things have changed really. I don't know if I answered your question.
Yeah.
But
I
try
to take an idea of your experience.
Yeah.
Anna, can you talk a little bit about the India variant for us, please?
So the double mutant that they talk, actually, one of the mutant that one of the mutation that he has present in this variant, that's the L452R, is the same variant that appeared before United States, and I just received a communication that some of this mutation appear here in sequence in Brazil. Not the same variant, because the Indian variant has a double mutation in the spike protein, and she's growing in India in the manner so fast that as appear in the mutant from Brazil, and he escaped from the immunological system too, because he has the A45484 key. Is a little bit different from the Brazil, that's a K, but it's also very important mutation. So he's spreading in India, and as far as I know, the curve of hospitalization and transmissibility is so high, but they didn't yet made some experiments in relation to the vaccine. But I think it will be the same that the other B17 for the England, South Africa, and Brazil, and the vaccine will continue to protect for this new variant. But it's a concern. I think she will be a variant of concern in a little time.
Yeah, and do you think that could be more interest for Brazil to try to put a severe barrier to the entrance inside of the country, could avoid this input of new variants or not? Do you think it's just a matter of time to have all these variants around the world?
I think it's important to have barriers in Brazil at this moment, because we are in a collapse for our health systems. So we have to deal with our variants and the variants that already arrived in Brazil, to have one more variant that has high transmissibility. We don't need this at this time. I think Brazil had to have more to impose more restrictive barrier from the outside country, as we happen in a lot of countries all over the world. And yes, at the moment, I think that is very important, because we cannot have a system from health that's worked very well, but is full at the moment.
And Maria, based on that cloud technology that you present today, we could see that the technology could be able to detect a lot of targets at the same time. And based on this, which is the cost of the reaction? Is it feasible to implement this kind of tool of technology based on public health or not?
Yes, indeed, it's implemented in a big number of hospitals here in Spain. And the cost, imagine, is a PCR, a tube of PCR, and the visualization process really is a microplate of plastic, and you print oligonucleotides in the bottom of the well. Really, the cost is almost less than 30% of the technicals that are competitors of our technical. Really, the cost is very good in this way.
Okay,
It's not
and
you don't use fluorescences, and you don't need equipment for analyze the fluorescences. Really, our equipment is a camera, take a picture, and we here develop the software which interprets the picture and offer the report. But the equipment is a camera, really.
okay. And Dana, do you think that we in Brazil, we need to sequence more samples? And based on these, do you think that could be interest to do active search in the population for new mutants?
Yes, we definitely we have to sequence more, and I think that at this moment we have a movement for different institutions to sequence more, and we are sequencing from patient. Dr. Akira asks, yes, we are sequencing direct from the PCR test. In my case, from the Rio de Janeiro, but also for different assays that is spread all over the country. And when we identify some new mutants, some new variants in some state, in some city, we try to focus to sequence more around the city to identify if this mutants is spread in this region, but we have to sequence more in Brazil to identify new variants, because they will appear, and with the vaccination, with the people having some vaccines, there will appear new variants. So we have to continue to sequence more genome in a fast way. Now we have some kits that is cheaper, they are cheaper, and we can produce in a little time, in 4 or 5 days. In 5 days, we can produce more or less 500, 600 genomes. So we have to have more sequence, definitely.
I'm totally agree. And one more question here. Dr. Ana Tereza, congratulations for the work. Do you think that vaccination could create new variants of concern from the selective pressure caused by the antibodies?
Yes, I think we will have some. I don't know if it will be a flock variant of concern, but will be variants of interest, of course, because we will have a pressure for the selection in this virus, and we will try to mutate his what the virus have to do, his work. So for that reason, I think we have to continue to work and to see which will appear. There is some projects ongoing that in that direction. One from coordinated by Fiocruz from the AstraZeneca vaccine that will sequencing the genomes in, I think it's Botucatu City, né? We will start soon. We will have one from the Butantan, from the Coronavac. So I think we will have some projects in this direction now.
Yeah, we are almost at the end of the session. I'm very excited with the discussion. I know that everybody's liking a lot, but if you want later, you could put more questions, and we could try to find a way to send to the presenters, okay? And thank you a lot, Ana and Maria, for the interesting contributions and presentations. It was amazing to be here today with you, and we appreciate the presence of the speakers and participation of the audience to contribute to discussions, and we invite all the participants to attend the next section. Thanks. Good afternoon.
Thank you very much, Maria
Bye.
Luisa.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.